Thank you, ISNTD, for the opportunity to talk to you today. So I'm talking to you about molecular diagnostics for schistosomiasis, but I just want to emphasize I don't come from a diagnostics background. I come from a molecular epidemiology background, and I've come into the diagnostics world looking at what kind of samples we're looking at, working in endemic areas and seeing what is feasible in terms of diagnostics and what's actually needed there. And that's really driven me to go forward and develop some of the diagnostics, which I'll show you here. So I'm going to briefly start by going through available diagnostics for schistosomiasis, the pros and cons of them. Then I'll present you some research which I've been doing using RPA, which was mentioned first thing this morning, an isothermal molecular diagnostic. And then I'll just round up by bringing together some final thoughts about diagnostics, and really thinking about differences and different diseases and why different diagnostics, same diagnostics won't be universal across diseases. So most people in the room will be aware of there's some very ambitious goals put forward for NTDs, and for schistosomiasis, there's some goals to eliminate schistosomiasis by 2025. And the main form of reaching these goals is through mass drug administration, and there's been some substantial donations, particularly from Merck, of praziquantel tablets to reach these goals. But there's a really immediate need for more sensitive and specific diagnostics to actually reach these goals and to really determine the true level of disease and transmission actually ongoing, because some of the transmission, some of the areas where we think we're reaching elimination, the prevalence might be much higher than we actually think. So this is adapted from Utzinger in Swiss TPH, and it really just shows that your diagnostic need changes as your disease level changes. So at the pre-control stage, you're really interested in mapping diseases, and your diagnostics really be high throughput at this stage. They need to be very, very low cost because you're screening large numbers of people. And your sensitivity and specificity can be slightly lower. And then as you move into control, you're controlling morbidity, and your diagnostic needs change again. But then when you start to get to that elimination stage, in the middle of the kind of phases of a control program, you really need really sensitive and specific diagnostics. And also you start to need to think about point-of-care diagnostics, because you really start to actually bring down to the detail of actually who's infected and who's involved in transmission. And you can see that those diagnostics, the sensitive and specific diagnostics, are still needed as you go through into transmission interruption, and then again at the post-elimination stage. So these are the main forms of diagnostics for human schistosome infections. So when I talk about human schistosomes, we're talking, these are mainly designed for the three main species, Mansenii japonicum, which would cause intestinal schistosomiasis, and then hematomin, which causes urogenital schistosomiasis. And it's also important to recognize that there's two forms of schistosomiasis. We've got urogenital and intestinal, and they manifest very differently, and the samples that you'll be analyzing are very different for those two diseases. So where we've got the ticks show how good those diagnostics are, and the crosses show where those diagnostics are failing. So we have microscopy, detection of eggs, and also the reagent strip for detecting blood in urine. They're very good at this mapping stage and for high throughput screening of people. The microscopy can be specific because we have egg morphologies, which we can look at to look at what species of infection. And they're also good in the field because they're low resource, they are rapid, they can be robust, and they're also very cost effective. So those are very good at that mapping stage. And then we have the antibody tests, which have been developed, and they vary, that their sensitivity can be much less, and their specificity actually reduces quite considerably compared to some of the other tests. But some of the antibody tests that have been developed can be implemented at the point of need scenario. And it's really the low cost and low resource diagnostics that are available really start to lose the sensitivity and specificity that is needed when we actually move towards this elimination setting. The most promising diagnostic probably coming through at the moment is the known as the lateral flow CAA test, developed by Leiden Universal Medical Center by Govert van Dam and Paul Korsenjans. And this is really showing great potential as an antigen test that can be used in field settings, but it still needs a lot of development in terms of the resources it needs, and it's also still very technically challenging. But when we start to use these antigen tests and we compare them to the microscopy tests, we start to see that our prevalence of infection is much higher than we originally thought. And this starts to really worry us, that we're actually, some of these elimination settings that we're dealing with actually have a much higher prevalence than we actually thought, or actually what we think we're aiming for. So I've just put together there that those are the key things that we're looking for in our diagnostics tests at those different phases in a control program. So phase 1 and phase 2, we have pretty much covered with those available diagnostics. And then we have, moving into 2 and 3, we have diagnostics which potentially can be used for the elimination, going into elimination. But molecular diagnostics really, as we heard throughout the day actually, that molecular diagnostics really offer that sensitivity, that high sensitivity and high specificity that we're looking for, especially when we start to move into elimination. So these are the molecular diagnostics which are available for us. We have qPCR, and we also have an isothermal assay known as LAMP. And you can see here that they offer sensitivity and specificity. But again, when you start to move them into the endemic setting and at the point of care need, they start to lose all the characteristics that we need to have in those endemic settings. So they're not ideal, and we really need to start to think about using molecular diagnostics and developing them and making them available in the field setting. So if you look at those molecular diagnostics, a lot of them have the specificity and sensitivity, but again, it comes down to cost, it comes down to deployment in those endemic settings, which is really starting to hold us back. But so these are the two main forms: qPCR, real-time PCR, detection of DNA through amplification, and then we have LAMP. But the LAMP assays are good in the field because they are isothermal, but they do start to lose specificity when they're actually used in the field. And it's important to realize that there's no one DNA target that's being used between diseases or even between species for the same disease. And these targets differ in their sensitivities, and there's no real uniform answer to which DNA target we should start to use. Also, when we think about sample types that we're looking at, so molecular diagnostics offer the ability to screen different sample types here. So for intestinal schistosomiasis, we can screen for eggs in stools, we can screen for cell-free parasite DNA in stool samples, urine samples, serum, and potentially saliva. I think there's been one study done on looking at saliva, which is a non-invasive sampling strategy which could have great promise. For urogenital schistosomiasis, we can look for eggs in urine, cell-free parasite DNA again. And we can also start to look at genital schistosomiasis as well, when this is a hot topic now with schistosomiasis, where we're having huge problems with genital schistosomiasis in women, but also men. And you can start to take samples from vaginal lavages or swabs in semen, and you can start to diagnose that kind of pathology of the disease using molecular diagnostics. So, and also multiple DNA targets can be screened using molecular methods. So different DNA targets can be used that are species specific, so you can look for multiple species. They can be multiplexed in assays, so you can do co-infection analysis. DNA targets vary in sensitivity, and you can start to, now that we've got a large amount of genome data coming out, we can start to screen these genomes for much more highly sensitive targets, especially designed around genomic repeat regions, and that can increase the specificity of molecular diagnostics. Assays can be quantitative as well, so you can get some idea about what kind of worm burdens you're dealing with. But again, the limitations come down to These assays are highly technical. They need infrastructure and resources, and those resources are mainly due to sample preparation, the actual running of the assay, the equipment needed, and also then analysing the data. So many of these point-of-care characteristics are not actually met yet. And again, the big hurdle is cost, because per sample, these assays are very expensive per sample. And currently, especially for schistosomiasis, they're really prohibiting their use in current control programs. So I've been working on a diagnostic known as recombinase polymerase amplification, RPA. And this is an alternative DNA amplification method. It's the same sort of principle as PCR, but it's isothermal. It's very quick, sensitive, simple. It can be low cost if you start to multiplex it, or you can start to split reactions. It's highly portable, and it's been shown to be very robust. And it's very good, and it's got very great potential at the point-of-care testing. So it uses special enzymes, known as RPA enzymes, to target DNA regions. And the sample you're dealing with doesn't need any particular manipulation, so you don't need high melting temperatures for your samples. And the amplification only occurs if the DNA is present, so it's highly homologous primer annealing. It's highly sensitive and can really amplify DNA from very low copy numbers. It's very rapid, takes 10 to 15 minutes, and it works from low temperatures, 25 to 37 degrees. And the way that the reaction works, it uses very low-powered battery devices. The actual chemistry and the enzymes are stored in a stable dried formulation, which can be taken out to endemic countries. And also, these enzymes are they're resilient to inhibitors. So, for instance, when you have inhibitors in something like urine or stool samples that are coming through after your sample preparation, these enzymes can withstand some of those inhibitors. And RPA is actually being used for multiple pathogens now, and there's quite a lot of literature coming out about that. So how do you detect your RPA reactions? Well, you can use it through standard gel electrophoresis if you have that equipment. You can have a standalone fluorescent detection. You can have real-time detection, and this real-time is a little portable machine that you can take out, and it's battery-powered. And then you also have lateral flow sticks, which can be used to detect your DNA amplification. So in collaboration with the London School of Tropical Medicine and Hygiene and through an MSc project, we worked on developing an RPA diagnostic for urogenital schistosomiasis. And the principle of this was based on either detecting eggs or free parasite worm DNA in urine samples. We decided to go for a target DNA known as the DRA1 repeat. It's a highly repetitive region in the genome, so this was going to offer us higher sensitivity. And from that, we designed RPA primers to target that DNA repeat. We analysed it through gel electrophoresis, and the assays worked. So the reaction takes 20 minutes at 37 degrees in a portable handheld device. And you can see on the gel there, we're getting down to 100 femtograms of DNA. So sensitivity was very good. We then redesigned our assay to work on a lateral flow system. And we have these lateral flow DNA dipsticks, which, again, the assay worked very well, and we had positive reactions here when 37 degrees and when the assay was run for 20 minutes. So again, this is a very nice kind of portable system to be able to use this and do a very quick analysis in the field. Again, we tested different serial dilutions, and again, we got down to about 100 femtograms of DNA. We then started testing different times and different temperatures of the assay, and actually the assay worked down at 30 degrees, and we did get some signal after 5 minutes of incubation, worked best at 10 minutes. So again, some of those reaction times will start to be reduced. And even if we're thinking about an endemic area, some of these countries are 30 plus degrees just in the natural air temperature. So some of you might not even need a heating device. We then, with this assay, we just tested it to see what happened when we actually put crude urine into our assay to see how it performed in the presence of normal PCR inhibitors. And again, it did withstand inhibitors at low levels of DNA. Inhibition did start to, when you start to increase the amount of urine, but at much higher levels of DNA, you can start to see there's no effect of actually adding those urine to the assays on the performance. So with another student from the London School, we actually went further to develop this assay into a real-time system. And this is using a portable battery-powered isothermal device with a fluorescent reader in there, and using the Lateral repeat. And again, we got, for a 10-minute reaction at 37 degrees, we got a real-time readout with our positive samples. And with the real-time RPA assay, we got much more higher sensitivity of the assay, 1,000 times more. We got down to 0.01 femtograms of DNA. So again, this starts to show potential for using this. The reactions are very quick, and they use low temperatures. And very recently, we've gone on to use this, develop these assays for schistosomiasis, the intestinal form, with a student, Kate Poulton, from the London School. Looking at 2 different DNA regions this time, ITS, 28S, using these handheld portable devices. And also, by the time we started this project, there was a newly available DNA lateral flow assay come out called PCRD, which is a much more user-friendly kind of lateral flow assay for detecting DNA amplification, and is much better for reducing contamination between samples as well. So for schistosomiasis, we got some nice results with looking at temperature tests. So we got positive reactions down to 30 degrees. We got reactions that only took 8 minutes. Sensitivity isn't quite as good as we'd like. We're only getting down to 10 picograms, but again, with further development, we can start to reduce that sensitivity, hopefully. So again, huge potential for that. So just a summary on RPA. It really does seem like it's a very good potential assay for point-of-care diagnostic tools. The devices we use are very portable. Sensitivity is good, but again, I think with more development, we can get much lower. Reactions are very quick. You need low temperatures. The assays withstand inhibitors. There's minimal sample preparation potentially needed, and some of the commercial companies that do DNA extractions are developing these speed extract kits now, which take they're very quick extractions and very crude extractions, which can start being incorporated into these kind of processes. The equipment that's used is very simple and field-friendly. And yes, as I said, I think with more development, we can start to increase the sensitivity and start to reduce any kind of false positive reactions that we're getting. So the unknowns with RPA, so this is all very much lab-based, and we need to start doing field testing, and hopefully by the end of the year, we'll be doing some field testing out in Zanzibar. Again, we don't know what to use as our gold standard to test these and to compare these assays to. We have egg output, we have some of the antigen tests, but we don't know what to compare it with, and that to allow us to see how good these assays are. We need to think about what sample type we're going to use and how we're going to prepare it in the field. And we also don't know what the sensitivity limits of those clinical samples are going to be, so that's going to be one challenging area. We also don't, in terms of sample type, when we're thinking about cell-free parasite DNA, which is probably the thing we want to go for because egg output in a sample is so random, especially when you get to low levels of infection. It varies from time of day to the number when you collect it, from one day to the next. So really targeting that cell-free parasite DNA within those samples is going to be key. But in terms of how that is in the system and in those clinical samples is very much unknown. So again, that needs some thought and some input. We need to look at what the best DNA targets are and start screening many more molecular targets. This image was shown earlier as well. So Dr. Ahmed Wahab from Germany is developing these mobile RPA suitcases, and he's been taking them around and doing diagnostics of many pathogens now, again showing that this is portable and can be implemented in the field. So now just to move back to molecular diagnostics for schistosomiasis and also ISNTD, so just to really highlight those major obstacles. So it's cost. And particularly for the RPA, they are costly. And again, too costly at the moment to incorporate into major control programs. Some of the solutions maybe are multiplexing, making multiplex pathogen platforms, trying to coordinate ways of sample collection and processing using existing projects or pathways, maybe developing sample pooling strategies to analyze large numbers of samples, reducing and cutting reaction volumes, so we're getting many more reactions out of what's actually considered one reaction at the moment, and considerable financial investment. Another obstacle is standardized methods. There's no clear consensus on which molecular diagnostic platform would be best in a control program, and there's no coordination about that. Some of the solutions maybe provide a platform that's most appropriate to the disease setting and needs and capabilities on the ground. Use standard quality assessment protocols and quantified reference materials to ensure robust comparable methods. Start to think about these non-invasive sampling methods, which will help compliance and participation of people in the, that we are testing, and also simplify the logistics of sample collection. Another obstacle is the result interpretation. So we have very little guidance on how to interpret these molecular diagnostics results and how we can confirm transmission, treatment failure, or success, or when we start to know when we're reaching elimination. And as I said, what are we comparing these diagnostic tests to? So we really need to develop good, robust guidelines and focus on intensity, probably rather than prevalence when we're doing these diagnostics. And develop focalized sampling strategies and also incorporate all the other data. We can't just look at what the diagnostics tell us. We need to see what the risk maps are telling us, look at what behavioral is going on in those changes, bring in something like vector xenomonitoring. And I'll just highlight Snell xenomonitoring there because my colleague Fiona will talk later on about monitoring vectors to look at monitoring of transmission. So just final slide. So it's really the priority characteristics for schistosomiasis to reach elimination. I think the most key thing is sensitivity and specificity of diagnostics, and also to develop point-of-care testing and high-throughput screening at that endemic setting. One of my worries is that when we start to use these sensitive techniques, the prevalence, which I think is very low, will actually be quite high. And in terms of schistosomiasis, I just wanted to finish by saying that when we're thinking about what our diagnostic tells us and what we can kind of get away with in terms of failures in diagnostics, when we're thinking about disease like schistosomiasis, if we have any false negatives results within our diagnostics, this really gives us a great risk of rapid reinsurgence. And this is really to do with the biology of the parasite. It takes one person to release some eggs into a water body. Those eggs will affect a snail, and within that snail we have rapid amplification, asexual reproduction. From going from one person that's come back as a false negative with a diagnostic can then reinfect a whole village or a whole population. And we can go from a situation where we've gone from almost transmission interruption right back into high prevalence again. So that's a real thing you need to think about when you're thinking about the pathogen you're dealing with, the biology of it, and what your diagnostics can do. And for that, I think point-of-care testing to deliver treatment rapidly to avoid contamination or non-compliance of treatment, and test and treat scenarios to really target those people that may be missing treatment or can start to lead into this resurgent situation again, because we could be back at square one before we know it. So with that, thank my colleagues, collaborators, and my students have done all this RPA work. The people at Leiden Medical Centre who often advise me on the background to diagnostics, people at TwistX, Ahmed in Germany, got a small grant from the Royal Society to do some of this work, and also funding through the London School Museum and also the Gates Foundation.